wanted to point out this particular array. So it's kind of a, you know, if you're not familiar with it, you might ask, well, why is that that way? This actually came out of Yuri Buzaki's lab. So if you're not familiar with Yuri Buzsaki, he's you know, a leading neuroscientist who's been involved with Michigan probes for many, many years. And he came out with this design, and now it's maybe our most popular design, which is really interesting. But what he does with it, this is the paper. I can send out the reference if anyone wants to see it, but I apologize, the figure's not showing up very well, but this is this probe here. And what he was able to do with that, because these are closely spaced, couple features of this probe. They're closely spaced. So let's see, for example, center to center here, you know, vertical center is 20 microns. So, you know, they're very closely spaced. And then with a known position of the electrodes, and the fact that the electrodes are on the edges, so there's a school of thought that believes that if you put the electrodes on the edge of the array, they're going to be closer to the tissue, closer to healthier tissue, and they're going to perform better. Nobody that I've seen has proven that, but it makes sense to me too. So with those features of this, he's able to do you know, spatial localization of neurons. And so this is a figure where each triangle is a neuron, because he's able to sample from multiple sites, so he can spatially localize the position of that neuron. And then he can determine whether they're functionally connected or not. So that the lines here, if you can see them, shows that, you know, like these two are functionally connected. And then the red triangles are inhibitory neurons, and the blue are excitatory neurons, or actually vice versa. But it's able to tell, you know, if they're functionally connected, if they're inhibitory, if they're excitatory, and where they're located. And then with that, in that same paper, you know, he showed some of the raw data. So here is the array implanted. It was an 8-shank array with 8 sites on each shank. And then they're color coordinated here. So shank 1 produced all of these traces here, and then 2, 3, and so on. But you can see the similarity in the signals, you know, within a given shank, you know, and then the differences between shanks.